A very good evening to one and all present here. Today, we will be discussing about a topic that is from food chemistry. We will be discussing about a topic that is from food chemistry, okay? And that is really important one, which uh, we can talk about. Yes. So what we will do is I will be talking about an important topic, and usually some gate questions also comes, competitive questions. So what we will do is we will discuss on this. That is production. Wait, 1 second. We will be discussing on this. That is production and well. So it is about production, okay, of glucose and fructose from starch. Okay, this is today's topic. Okay, now why I have chosen this topic? It's a very basic topic for the food, from food chemistry topic. Okay, we usually get some questions for gate for this. If you say, we usually have some gate questions from here. So I have taken this topic in order to just a few points I need to clarify here. Along with that, you can also refer some books like, that Shakuntala Manai, then Shiva Shankar, all these things, V. Shiva Shankar, like some food chemistry books, even Panama. You can definitely refer those books. Here, I'll just give you some points so that it will be easy for you to get the basic idea how we can produce this. The lecture is mainly intended for the BTech food technologists as well as for the diploma for food processing and food technology, that is polytechnic colleges and BTech level, UG level. Okay, so let's start the discussion. So first is starch. Okay, how and what is happening for starch to glucose? How will you convert the starch to glucose? Clear. So this first thing, when I'm saying there are three methods. The first thing we will be talking about is gelatinization. The first thing is gelatinization. Okay. The second thing is about the liquefaction. Okay. And the third thing is about saccharifaction. Yes. Saccharification. Okay. So whenever there is a question like that, how can you produce the glucose and fructose from starch? So when we need to convert the starch to glucose, we will be taking the three major steps. First is the gelatinization. Second, the step is liquefaction. Third is saccharifaction. Okay. Now, in short, if I tell about gelatinization, say suppose we are having some cornstarch. Corn starch. We have cornstarch. Okay. We have potato starch and some of the starches from grains. We can take cornstarch. We can take the starch from potato and some other grains. Okay. Now, the basic idea you are aware of, that is starch will usually not dissolve in what? What is the thing? It won't dissolve in water. Okay. The starch do not dissolve in water. Do not dissolve in water. But at the same time, it disperses. Why I'm focusing on this? Because it will be important for you to know if there is like the starch usually will not dissolve in water, but it will disperse and become an emulsion kind of thing. So it gets dispersed. The starch won't dissolve. This is an important key point you can note. Similarly, so the substrate needs to be dissolved in water. Now, whenever we are taking the starch, usually what happens? The starch usually disperses in the water. So the starch disperses in the water, as I mentioned, and what happens? The swelling takes place. It swells up. Okay, ultimately dissolves. That is called gelatinization of starch. So I will come back to the points once again. It will be easy for you to understand what all, like I am talking about how we can produce the glucose and fructose from the starch. So in order to convert the starch to glucose, I have pointed out the 3 points here. That is, first step, gelatinization, liquefaction, saccharifaction. Okay, saccharification. Now, I mentioned one point here. You are aware of like what are the sources of starch? Mainly the examples like corn starch, potato starch. Okay. Then we have starch is basically do not dissolve in water, but it disperses in water. So at a particular temperature, usually if you tell about the gelatinization temperature, what happens? Different starches. If I talk about cornstarch, the cornstarch is having around 60 to 72 degrees Celsius in which it gelatinizes. Okay, that means when I'm dissolving in water, the starch won't dissolve. Then what happens? The starch is dispersing in water. When the starch is dispersing in water, it starts or it has a tendency to swell. When it is swelling, then what happens? As a result, finally it gets dissolved and the particular term is called as, after swelling, the particular term is called as gelatinization of starch. Okay. So usually the gelatinization of starch takes place in certain cases. If you talk about bread baking, it will take place in gelatinization of starch takes place in that too. Okay, so one such application is like, it can be, you can say about parboiling. Parboiling is an application, or you, okay, one thing is can be told as in baking, the gelatinization plays a role. So how do you relate all the points? I can relate the points like starch. Why I have mentioned here as 60 to 72. If I am taking the cornstarch, then only the particular gelatinization temperature is 60 to 72. But if I take for wheat, definitely if I am talking about wheat starch, then definitely there will be, if I'm talking about wheat starch, okay, so definitely what happens, there will be, the temperature will be around 58 to 64 degree Celsius. So at that temperature only it will be start to gelatinize. My suggestion is for few starches, like I have mentioned one corn, then for wheat, then if you want you can even talk about potato. This potato, basically the starch content will be around 59 to 68 degrees Celsius. Okay, so for wheat, for potato, for rice, for corn, for barley, if you remember this data range, it will be easy, because sometimes there are some questions in the competitive exams that will be helpful for you. Okay, and basically the normal temperature which we can say usually at what time it gelatinizes. So it gelatinizes at around 80 degrees Celsius, we can say that it gelatinizes, and as a result, you will be the start gelatinization. Okay, 80 to 82 range also you can mention. Now the next point, which now I will erase this part, and next topic, next part I will be discussing with you is regarding. Yes, I'll just erase this portion and come back to the next part. The next part will be dealing with. Now you know what? I think you have studied. I think you have. You know what? Before that, when you have started the topics, I think regarding starch you have studied. Yes. If you have started regarding starch, then definitely you must be aware of the two types. One is amylose, another is amylopectin. Okay, so this will be, it was, might have been detailed studied. You know this concept, or if not, I'll just give a small quick difference. You can say this is having a branch chain, amylopectin, and this is the amylose are having the straight chain concept, straight chain. Okay. Now, what can I say here? So basic matter is amylose will be usually in the starch around 20 to 30% content will be amylose, and the branch in amylopectin will be around 70 to 80% is present. So this particular thing you have to again remember. So starch, the key points which you can take away points are like starch is not dissolving in water, then starch disperses in water. Starch is of the two are main, like we can say amylose and amylopectin in this particular proportion constitute and make up the complete starch. And starch in the two ways, that is amylose and amylopectin. Gelatinization of starch for the different types which I discussed, maybe cornstarch, potato starch, wheat starch we have discussed, and the common gelatinization temperature. Okay. Now we will be talking about one enzyme cannot act on any if the starch is not gelatinized. The enzymes won't be able to act on that. Okay. We need to gelatinize the starch so that the enzyme, for the enzyme to act, we need the if the enzyme in order so the enzymes are acted upon, we need the enzymes, so we need to gelatinize the starch. Okay, so starch needs to be gelatinized in order to for the enzymes to act upon. This is another point. Now the next point we will be talking about is dextrose equivalent. What do you know by what do you understand by the term dextrose equivalent? This is a very common and sometimes a hot favorite topic for questions. Okay, so you will be please know what is dextrose equivalent. Dextrose equivalent is nothing but you can say it will be defined. How can you define it? It is defined as reducing sugars. Okay, the reducing sugars which is expressed in as dextrose. The reducing sugars that is expressed as dextrose. Okay, the reducing sugar that is expressed as dextrose, is it okay? And calculated as percent of dry. So the reducing sugars which are expressed as the dextrose, and that will be expressed and calculated as a matter of dry substances. That will be taken as your dextrose equivalent. Okay. The next thing is what we will do is we will be going for the next part. The next part which we will be dealing with is about the how do you hydrolyze the starch. Okay, so hydrolysis of starch can be one another part. Let me omit this and I will come back to that. Hydrolysis. So what are two types you can say? One is acid, another is acid followed by enzyme. Okay, and another part is enzyme hydrolysis. Okay, there are three types of hydrolysis: acid enzyme, as well as only enzymatic enzyme that you can say as enzymatic or enzyme hydrolysis. Is it clear? So now what I will do is I will be talking about this, like say suppose you are having the acid part. How do you do acid hydrolysis? One such method we can say like we are using it for a long time. The starch is really readily or will be dissolving or hydrolyzing by the dilute acids. So we can do what? In order to hydrolyze that, we can use the dilute acids. Okay, in the dilute acids, the solution gives quantitative, it gives the amount of glucose. So with the help of acids, dilute acids, we can hydrolyze the starch and the amount of glucose will come. But what is the disadvantage here? It is giving some problems like in imparting bitter flavor, it is corrosive, impart bitter flavor, okay, right? If the extent of acid hydrolysis is more, as well as what happens is it is, and the dextrose equivalent, that is, I mentioned you in the before, just before this, we took the topic, that is dextrose equivalent, right? That is the number of defined as the reducing sugar, which is expressed as dextrose. So the dextrose equivalent is also very not more than 55. It is not greater than equal to 55 we get. This is the problem with acid hydrolysis. But it is a conventional method or which has been used from a long time. That is nothing but the starch will be used in the presence of dilute acids, right? In the dilute acids, and that will be getting hydrolyzed to form the glucose. But it imparts some bittery flavor, as well as the dextrose equivalent is around 35. Sorry, it will be around not more than or equal to 35. Okay. Next, we will be talking about your next topic, that is about how do you go for enzyme acids followed by acid enzyme hydrolysis is the second step. In the second step, if you are talking about acid enzyme hydrolysis, over there I will be telling you about how this is the second step we will be discussing with. Okay, acid enzyme. If I'm talking about acid enzyme hydrolysis, what can we do? Acid enzyme is like first, the acid hydrolysis will be your first step. So this will be your step 1. In step 1, what do you do? Acid hydrolysis, we will get 18 to 20. 18 to 20 we will be getting around 18 to 20 we will be getting around the dextrose equivalent for step 1. The same procedure we will be following. Yes, this is the dextrose equivalent we are getting. Followed to, then we will do enzymatic hydrolysis. Okay, so the acid hydrolysis followed by enzymatic hydrolysis will be done here. Acid enzyme. This is your second method. Okay, and the third method is about the enzyme hydrolysis. What do you do in this? The liquefaction will be done by some enzymes. When we talk about liquefaction, this will be done by the enzymes. Okay, what type of enzymes it will be doing with? The enzymes that like thermostable bacterial alpha amylase. Why thermostable? Because the bacteria, this we will be getting from thermostable bacterial alpha amylase. Okay, so this particular, so here the question is like, if you are going for a liquefaction process, then what enzymes like alpha amylase is involved, followed by you will have the saccharification process, which will be done by fungal amylase. Okay, saccharification we will do, that we will be getting, this we will do by fungal amylase. Okay, and finally we will be getting the glucose. Is it clear? Now my point is, why we are discussing all this here. Just take the key points here, that is acid enzyme. What is the step 1? How much dextrose equivalent they get? This you can you need to remember. Similarly, when we are doing the enzyme hydrolysis, okay, what do you do? The liquefaction is done by bacterial thermostable bacterial alpha amylase. Saccharification will be done by the fungal amylase, and we are getting crystalline glucose, which is obtained. Is it clear? So there will be certain parameters in order to follow this particular when we are converting doing the hydrolysis. Such parameters will be of pH. It will be the time, temperature. Okay. Then the enzyme, the presence of the stabilizing agent, calcium. Say suppose, what do you understand? Suppose for alpha amylase, you may have a stabilizing agent like, the stabilizing agent is calcium. Okay, Ca. What is that? So this is the thing what we can discuss about the basics of different type of hydrolysis method. Next is I hope till now you are clear with it. So basic things what we have discussed about how to convert glucose, how to get glucose and fructose from the starch. What are the three steps? Gelatinization, liquefaction, and saccharification. Then how do you go for the, like what is dextrose equivalent? Then what are the types of hydrolysis system? Like the types, mainly we can have acid hydrolysis, we can have acid enzyme and enzyme alone. Okay. So this particular parts we can, the three methods we can use, and what are the enzymes involved in that mainly. You should know about it. Now, if I talk about a particular, say suppose I'm telling, since I already told you what is this particular gelatinization and liquefaction part. Now, I will be coming with a basic idea regarding what exactly happens in the basic process. Okay, very simply, I will tell you, which will just give you an idea how it takes. Say suppose I am talking about starch, gelatinization and liquefaction. How does it take place? Starch, gelatinization and liquefaction. Okay. So what do you understand by this? When we are doing gelatinization and what happens is the liquefaction. The starch may be slurried in water. Okay. We are taking a slurry solution. And when we are heating it, it is just getting, suppose we are taking like this one starch solution. The starch solution will be in a slurry form. Okay. The starch solution is in the slurry form. Okay. Now, when we are heating it, it will be resulting without any enzyme. It gives you a colloidal solution because why? You will get a viscous and colloid solution here. Okay, starch solution. If we are not adding any enzyme here, we are not adding till now, no enzyme. So the starch solution is already in a slurry and it is viscous. It forms colloidal colloids. Okay, like a colloidal solution it becomes as it is extremely viscous at higher starch concentration. Okay, so we need to add some amount of enzymes before heating. When we are heating this starch slurry, it is giving you some colloid type of solution because of its extreme, because of the viscosity, increase in the viscosity of the solution, leading to the colloid structure. Okay, when we are not adding any enzyme. So for this, what we are thinking we will do is, if we add an enzyme at the heating period, the starch will hydrolyze at the moment of gelatinization. So the idea of speaking this is, if you go, when the starch is, when an enzyme is added here, when I am adding an enzyme here, the starch will start to hydrolyze. Okay, rather than they will start to hydrolyze during gelatinization rather than getting into a colloid structure. This is what I was supposed to mean here. Okay, so the fungal alpha amylase won't be sufficient, okay, to survive at the gelatinization temperature, because we have already discussed the gelatinization temperature in a range we can take approx 80 to 82, followed by the different type of starches will have different type of gelatinization temperature. Okay, but if we are generally taking around this, the fungal alpha amylase won't survive in that, right? And it will be maybe so. What we are doing is, we will also add alpha amylase that may be used, but inactivation temperature is closed. Okay, so to the gelatinization temperature, we can use fungal won't be that helpful. We can even use alpha amylase. Okay, we can also use alpha amylase for this particular work. I mean the starch gelatinization. In contrast, some bacterial and alpha amylase which are stable, we can also use them, okay, for liquefaction of starch. So for liquefaction, we can use usually the fungal will be usually cannot withstand the gelatinization temperature. Alpha amylase we can use, and even some thermostable alpha amylase or which is obtained from bacteria, which I have discussed in my immediate previous discussion. Over there also, I have said the thermostable bacterial alpha amylase that can be used. Okay. Now, alpha amylase we can obtain from, for example, one such example is Bacillus licheniformis. Licheniformis. We will get it from alpha amylase. I have given you this name because even amyloleucophacians, Bacillus amyloleucophacians. Why am I talking about this? Because this type of alpha amylase we can get from the bacteria. So from the bacteria we can get this type of alpha amylases which we can use for the liquefaction process. So this is what I wanted to mean here. Okay. Then the next idea about this is your the next idea is about the starch liquefaction. We were telling about in terms of gelatinization. Then if I am talking about the liquefaction, okay. So liquefaction, what do we do here? When I'm talking about liquefaction, so mainly for liquefaction, the pH should be around 6.5. Okay, and the starch concentration you need 30 to 40%. 30 to 40% of starch concentration can be taken at a pH of 6.5. The liquefaction starts. So if the alpha amylase, I have already mentioned just immediate before, the alpha amylase can be obtained from the bacteria like Bacillus licheniformis and Bacillus amylophaciens. So if I am talking about if the particular alpha amylase which we are using for liquefaction is from Bacillus amylophaciens, the enzyme will be added at the beginning. If you take alpha amylase from Bacillus leucophaciens, amyloleucophaciens, okay, from this bacteria, amyloleucophaciens, if this is taken, right? If this is the part which you are taking the alpha amylase for liquefaction, definitely the enzyme added at the needs to be added at the beginning at 80 degree. Beginning of the beginning you need to add it at the okay, gelatinization at the beginning we need to add, and we need to keep it for around maybe 1 hour to 1 hour 15 minutes or so. Okay, then we have to raise the temperature to 140 degrees Celsius under pressure. Okay, under pressure or vacuum we will be raising the temperature to 140, right? And then we are cooling it to 85. So more enzymes added to the liquid, and at this temperature, so we can add, when we are cooling, we can again add some more enzymes. Okay, and this can be taken and held for another time. Now, what is the, since I have discussed with two parameters, that I mean two bacteria alpha amylase can be obtained. One is Bacillus leucophaciens and the other one licheniformis. Another one is Bacillus amyloleucophaciens. Okay, so when I have discussed about these two, definitely there will be a question like which one is more thermostable. You should know that. So the Bacillus, the alpha amylase, which we obtain from the Bacillus licheniformis, is more, okay? It is more thermostable, and you can use it at once, right? And similarly, the dextrose equivalent we will be usually get as 8 to 15 dA. That means the Bacillus licheniformis is more thermostable. The alpha amylase which we obtain from Bacillus licheniformis is more thermostable, and we can use it once, Okay, beginning, you can add it at the beginning and you can raise the temperature till 105 degrees Celsius. And we can keep it for 5 minutes and just bring it back to 95. Cool it to 95 degrees Celsius. Okay. For around 2 hours, we can keep held for 2 hours. So the process of hydrolysis, we will be getting this 8 to 15 B I have mentioned here, no? Dextrose equivalent we will get in this hydrolysis process. Is it clear? Now, the next part we will be discussing about is what is saccharification. So if we discuss about the saccharification, so this saccharification is nothing but what is saccharification? It will be, I mentioned there, right? It can be 1 minute. Okay. It can be carried out by fungal alpha amylase, okay? Or in combination, you can go what? High maltose syrup. So if I'm talking about saccharification is like we will be having that is carried out by alpha. So alpha fungal amylase, if it is there, we can carry out the particular can be carried out by fungal alpha amylase. Okay. Even we can have another one that is fungal glucosidase. So with this enzymes, we can carry out the what we can do. We can carry out the saccharification process. What is the basic mechanism behind this saccharification process? The mechanism behind saccharification processes, they will break the 1,4 linkage of starch. Linkage of, they will break the alpha 1,4 linkage of starch. Okay, that is alpha amylase. And alpha glucosidase, they will break alpha amylo glucosidase. They will break the 1,4 linkage and 1,4 and 1,6 linkage. Now, you may be thinking, why this much of notation, why this much of writing and all? My funda is, why I am showing this to you. Saccharification mainly is done by alpha amylase, that you may know the two enzymes here. This is my first point to take this. What is the mechanism? They will be breaking the 1,4 linkage and they will be breaking the 1,4 and 1,6 linkage in the saccharification process. For amylose, it is 1,4 linkage of starch, and for amyloglucosidase, it is 1,4 and 1,6. So this type of questions are there often in GATE. Please do remember, like which linkage is they are breaking, what is exactly happening in saccharification, why hydrolysis, what is the gelatinization, importance of gelatinization. These parts we can definitely go with. Now, this is about saccharification. And in the saccharification technique, what I can tell you about is you can keep, like, in commercial process, we can use another thing is commercially. Commercially, we use mainly even the dextrose. Okay. Even what I'll tell you, the saccharification, as I mentioned there, the liquefaction takes place at a pH of around 6.2, correct? That also I mentioned you around 6.2 or 6.2 around 6.5, that I have mentioned before. Saccharification will be definitely at a pH of around 4.5. It will be taking place with a temperature of 60 degrees Celsius. Maybe we can say around to 24 to 110 hours. This can be the duration depending on the experiments what you are doing. So the slide, so what happens, slight degree of agitation we need to give in order to go the, the expedite the process of saccharification. So when we are getting the, in the commercial process, what happens? The saccharification commercially we are giving, when we are doing saccharification, we are getting finally the glucose, right? So this glucose in the commercial level, it is called as dextrose. Commercially, they will be saying this much amount of dextrose, and they will be sometimes expressing that also in terms like, you know, I told you before also, it is in commercially like dextrose equivalent, what is the definition? So in commercially, they are like after saccharification, it is expressed as dextrose instead of glucose, and it will be produced like how much DE, dextrose equivalent, they are having in the like common form, we are getting it as dextrose monohydrate. That will be produced in the DE, dextrose equivalent. How much is it? Mainly, it will be coming around 95 to 98 dextrose equivalent after the saccharification process. Is it clear? Now, the next thing what I'll be dealing with is your like see, what happens is this much is there, and then when you are getting this dextrose in the form of dextrose, we say, right, then goes with your, so we have completed till saccharification where we are getting the glucose often called as dextrose commercially. This glucose often called as dextrose commercially will be further going for isomerization take process or glucose isomerase. Okay, in isomerization, that is glucose isomerase. So you will be getting as fructose. Okay, is it clear? So when and followed by, we can even go for your high fructose corn syrup, which we usually obtain as 42% HFCs and 55% fructose concentration. So these high fructose corn syrup will be another different topic which can be dealt with in the later part. So this is about your topic today, what I was about to discuss. That is, how do you convert the starch into glucose and fructose, followed by the major 3 steps. Okay. So in a nutshell, I will just summarize my lecture for this session. What all we have discussed today in this lecture, just I will put it in a nutshell, and with that we will finish. So first of all, I have discussed about how can you convert the starch into glucose and fructose. This was my topic. So from starch, we have 3 processes. Okay, hydrolysis, liquefaction, saccharification. Okay, hydrolysis mainly with the gelatinization part. Okay, gelatinization part, then the liquefaction part, and then the saccharification part. So these 3 we have discussed. Then what are the gelatinization temperature? What happens in gelatinization? That mechanism of what happens in gelatinization. This was what we discussed. Then what is liquefaction, at what pH and temperature, what are the enzymes involved? And in this hydrolysis method, what are the types you have? Acid, we know, acid, enzyme we know, and the enzyme we know. Okay, so these all will be there in that part. Then what are the enzymes responsible for liquefaction? Same with the saccharification. What are the time temperature constants? And what is main thing is what is dextrose equivalent? What do you understand by that? Okay, then we will be going with saccharification at pH, and then from the last step, that where we got glucose. The glucose is often called as dextrose and commercially. That part we have to deal with. And finally, this glucose will be isomerization with the presence of glucose isomerase will convert and give you the fructose. From there, even we can go for the high fructose corn syrup production. That can be done as one part. So all these things we have discussed today. And with this, I would like to conclude the session for now. This, whatever I have highlighted the key points, that usually is a hot favorite part where you can get some food chemistry questions in certain competitive exams. Just focus on that. This was just a nutshell of what we can do. We can have a overview of this particular topic. Thank you. This lecture has been taken by myself, Pailas Gupta, Research Scholar, Department of Agriculture and Food Engineering, IIT Kharagpur. Thank you so much.